Hello everyone, welcome back to Biology Bites. So today we'll discuss the concept on the important enzymes and their roles during DNA replication. So coming to the enzymes, see these enzymes, they play a major role in the DNA replication. We know that the replication of DNA is semi-conservative mode. Why it is called semi-conservative? Because half of the strands will be having a parental and the other half of the strands will be having a features of the new strand, that is a daughter strands. So now during this semi-conservative mode of replication, there are few machinery enzymes that are involved to carry this replication. And the type of the replication that is formed is inverted Y shape. How this inverted Y shape replication is formed? So what are the enzymes that are involved? The first enzyme is DNA polymerase. This DNA polymerase, poly means what? Helps in synthesizing more number of DNA. Helps in synthesizing new strands of DNA by adding what? By adding the nucleotides to the new strands of DNA. So that it can maintain a proper cell division. A proper cell division is seen and the proper replication of the genetic, the gene information also is seen during the cell division. Helicase. Helicase means what? Helix. So we know that the DNA it is of helical shape. So now this helicase enzyme will unwind the DNA. Unwind means what? It will open up the DNA strands so that it allows a replication properly, allows a proper replication, or the replication mechanisms will be checked by this helicase enzyme. Topoisomerase. So what this topoisomerase will do? So now just in case, for example, now if you take a thread, just take a thread and just stretch it and leave it. When you leave it, what will happen? The coils will be formed. To prevent that coils, the topoisomerase plays a major role. So prevents the DNA tangling during replication and helps in maintaining a genome stability. Ligase. Right. This ligase is called as DNA stitches. The ligase is called as DNA stitches. Stitches means what? Joints. It's joints which fragments Okazaki fragments. So during replication, you will see a small fragments that are formed, that is called as Okazaki fragments. Those Okazaki fragments are the DNA molecules or DNA fragments to maintain the continuity. That means all the nucleotides will be properly aligned and formed in the DNA, ensuring a complete gene transmission. So that is the use of DNA ligase. Then coming to the primase. Primase, it is only for the synthesis of RNA primers. Basically, plays a major role during the mRNA synthesis. Right? Initiates DNA synthesis at the correct location. The synthesis of DNA that should take place, that is checked, and the synthesis of RNA that is checked by primase enzyme. REN. REN is considered as molecular scissors. REN is considered as molecular scissors which identifies a specific sequence of DNA. So if you take this nucleases, nucleases is the name of a class which is further divided into exonuclease and endonuclease. Exo and endonucleases means which identifies a specific sequence of DNA, and it is very much useful in the recombination. Especially this plays a major role during the biotechnology, so that the new genes, that is the foreign genes, will be combined with a vector gene so that it helps in formation of a new forms of DNA that is called as rDNA. Then recombinase. Recombinase helps in crossing over. So that means what is that? During this meiosis 1, you have studied the different stages that is in the meiosis 1, that is prophase 1, metaphase 1, anaphase 1, telophase 1. Again in the prophase 1, we have studied leptotin, zygotine, pachytine, diplotin, dikinesis. So here during this meiosis stage, you will see the involvement of paternal and maternal genes. The genes of both paternal and maternal will be crossed, or will be crossing over takes place. Once when the crossing over takes place, the recombination of the genes will takes place. So that is done by this recombinase enzyme. The last one is reverse transcriptase. So this reverse transcriptase So make a note on that. This is found only in virus, especially in HIV virus, right? So why it is called reverse? Usually, what is the definition of transcription? Synthesis of what? Synthesis of mRNA from DNA. But in the reverse transcription, what is synthesized? DNA is synthesized from RNA. DNA is synthesized from RNA. So here, this particular type of reverse transcription is found in this HIV. Clear? So these are the enzymes that are involved in the replication part of DNA. Hope all these concepts are clear. Now, we'll discuss few MCQ questions based on this enzymes. Coming to the first question. During DNA replication, if the activity of DNA ligase is inhibited, which of the following will occur? Here they have asked DNA ligase. So DNA ligase means what? Ligase major, what is the function of DNA ligase? Helps in combining the fragments. So here, do you find any fragments in the first option? No. Coming to the third option, the DNA polymerase helps in getting multiple copies of DNA. Replication fork will not be formed by DNA ligase. So that means the option will be B option, Okazaki fragments. So here the ligase joins the Okazaki fragments on the lagging strand. Without it, small DNA remains unjoined. So that means the option is second one. Coming to the next question, which enzyme prevents the DNA double helix from getting overwhelmed ahead of the replication fork? So which enzyme? DNA ligase? No, DNA ligase helps in combining what? The Okazaki fragments. Helicases helps in unwinding the DNA. DNA polymerase again helps in multiplication of DNA, but topoisomerase. Right? So what this topoisomerase will do? It will remove the supercoils. It will remove the supercoils and tension during the DNA unwinding. That means what will be the option, students? It will be second option. Then coming to the next question. The enzyme that initiates DNA synthesis by laying down a short RNA sequence is primase. Why it is primase? Here initiates DNA synthesis by laying down short RNA. Make a note on this point. Short RNA. DNA polymerase is for DNA. Helicase is unwinding the helical structure of DNA. Ligase is for joining the DNA fragment. So that means what will be the option? Second option. So primase synthesizes RNA primers to start DNA replication. Which enzyme is directly responsible for ensuring genetic continuity during cell division? They are asking enzyme directly responsible for genetic continuity. Recombinase? No, students. Recombinase it is not. Recombinase is only during what? Meiosis. And coming to the restriction endonuclease, yes, this is involved only in the biotechnology, helps in fusing the foreign gene and the vector gene. RNA polymerase, no, absolutely it is wrong. So the genetic continuity is maintained only by which enzyme? DNA polymerase. Clear? So this DNA polymerase copies the genetic material precisely to ensure inheritance from parent to the offspring. Oh, the concepts and the MCQs are clear. Thank you.